Welcome to Shellphone, the podcast that gives the ocean its very own hotline. Join us as we hear from ocean stewards, discover threats to ocean health, and learn ways we can all answer the ocean's call. This podcast is brought to you by Breach the Surface and Coastal Creative. I'm Harmony Dawson, and joining us this episode is Melody Lindsay, a geomicrobiologist from the Bigelow Laboratory for Ocean Sciences. Hi, Melody. Thanks for being here today. Before we get started, can you give us an introduction into your background and the work that you're doing now?
Yeah, of course. And yeah, thank you so much for having me. I'm so excited to be here today and to talk with you about my research as a geomicrobiologist and also an astrobiologist, which I'll get into a little bit. So my background working in ocean sciences right now at Bigelow Laboratory for Ocean Sciences is not in ocean sciences. I grew up in Hawaii, but wasn't really interested in ocean sciences, but what I was really interested in was astronomy. And I was really interested in the question, are we alone in the universe? Kind of the astrobiology question, is life possible elsewhere? What does that life perhaps look like? And so I was really interested in this field in particular. And so in my undergraduate research, I joined a geomicrobiology lab that was looking at life that was living in the deep subsurface in a gold mine. And so they live in these underground lakes, and they could be really similar to environments you might find on Mars or on Europa or on Enceladus, kind of these extant worlds. And so that's how I really got into the geomicrobiology and the interactions between rocks and water chemistry and how that provides energy for life to grow in the subsurface. With that work, I started to look at hot springs in Yellowstone National Park, kind of these expressions of the subsurface on the surface. Those water-rock interactions in the deep subsurface aquifers, the water beneath Yellowstone, is completely impacted by the type of rock that's there, the hydrothermal activity, and that fuels microbes and the activity of microbes in those hot springs. And so that was my PhD work at Montana State University. After that, I came to Bigelow Laboratory in order to study the deep oceanic subsurface. And so the environment beneath the ocean can be quite similar to the environment beneath the surface of land. And so the microbes that live there have common metabolisms. They use a lot of sulfur and stuff like that. And I was interested in seeing what microbes are there that are active. What are they actually contributing to different global biogeochemical cycles of different compounds like carbon, sulfur, nitrogen, stuff like that. It's these really universal microbial metabolisms that we don't know a lot about because they're difficult to access, being either deep in the ocean or underneath the ocean or deep in the subsurface. And so that's kind of where my research is focused today, is looking at activity rates of microbes that do metabolize these chemical compounds in ocean sediment, beneath ocean sediment, but also I still do a little bit in the terrestrial world, the land-based world, in the deep subsurface there as well. All of this to say, geomicrobiology work is really tied into astrobiology interests in seeing what these environments on Earth can tell us about environments elsewhere in the universe. And are we alone?
Awesome. That sounds super interesting and is a world that I haven't really dove into, but after researching for this episode, I'm all about it and I want to learn a lot more. My first kind of quick question is, we already kind of got a sense of it from that description and I think the word itself, but what does geomicrobiology mean?
Yeah, great questions. Yeah, so geomicrobiology, and there's a lot of different facets of geomicrobiology. A lot of geomicrobiologists kind of edge into the realm of biogeochemistry, and so it's really this geomicrobiology. You can separate into the geo, which is the geology, and so rock hosted, and then the microbiology is all about microbes, and so microbes that live particularly on rocks or live on chemicals that are derived from water-rock interactions or sediment water interactions. That's what geomicrobiology focuses a lot on. And so it can really range from anything from the microbiology part of it to the chemistry part of it, those interactions between microorganisms and chemical cycles, and also the geological processes. Another part of geomicrobiology is kind of studying the process of how life came to be on Earth. And so that's more maybe geobiology, so the origins of life on Earth, and where that came from in terms of how did RNA and DNA molecules come to be? Did that have something to do with the interaction of physical, geological processes and microbiology, and how that evolution happened in the 3.5 billion years of life on Earth? So that's another part of it, but not one that I'm too into. Not I'm into it, but not one that I'm really involved in with my research.
Yeah.
Yeah, currently. Yeah.
What are the important things to know about microbes? Like, what are they? How big are they? Where do they live? Anything like that that's would be a basis for the rest of the conversation.
Yeah, of course. Yeah, what are microbes? That is a great question. And I don't think microbiologists sometimes have a good handle on what is a microbe. So a microbe is really just a tiny organism. When I think of microbes myself, the ones that I study in particular are single-celled bacteria and archaea. Some people will call bacteria and archaea together prokaryotes, which contrasts with eukaryotes, which is what humans are, all mammals, plants. But there can also be eukaryotic microbes. So like what is the cutoff there? So there's maybe a couple different definitions of those. But so I think microbes are just really small organisms. They can really range in size anywhere between a viral particle, which is really small. If we think of a human size, a human cell, that's a eukaryotic cell is about 10 times bigger than a microbe. And a microbe can be anywhere between zero point zero 0.01 microns, which sense of scale is a little off, to 10 microns. And then viruses can be 10 to 100 times smaller than that. So they're all really tiny. You won't be able to see them with just your vision, but with a 1000x microscope, you can certainly see sometimes even the smallest ones. And so that gives maybe a sense of scale to some degree, but it is hard to conceptualize how tiny they are in general, especially via my audio description. But these microbes are very important to the earth and to humans in general. Like if you think of your microbiome, there are more microbial cells within each human than there are human cells within each human. They're just that much smaller, so they actually don't take up as much volume as the human cells would. But that's the same way that they function on the earth in general. There's a huge amount of microbes everywhere. They represent the majority of life forms on Earth, maybe not by volume, but by sheer number of them. They live everywhere. You can't escape them. There's very few environments on Earth where we find low biomass microbial life and no biomass microbial life. One of those being like in an active volcano, just because it's so hot. That's like one of the few places a microbe can't live. So they're really just super widespread. Sorry, was there anything else? I tried to give a pause. Was there something that I missed? I guess anything important.
I think you covered all the specific
Okay,
points
great.
that
Yeah,
I asked about.
sweet. Yeah,
Yeah.
great. Okay.
And that's really interesting to hear about like volcanoes and where you wouldn't find microbes when they're so all over the place.
Yeah, I mean, the microbes, there's aeromicrobiology even, so microbes that live in the atmosphere and they can like seed clouds and cause rainstorms, and they do a bunch of chemical cycling up in those kinds of environments. So really, even up until like the stratosphere, getting into outer space, there's probably microbes there as well, doing their thing.
So a lot of your work has to do with the genetics of microbes. Can you give us an introduction into what genetic diversity looks like in microbes, particularly the ones that you study, and how you study that?
Yeah. Yeah. So microbes are incredibly diverse, both in genetic diversity and also what they're able to do as a result of that genetic diversity. And so microbes are, I think, really special, especially the microbes that I study, because they're able to do a wide variety of metabolisms. And so you and I, as humans, we respire oxygen, we breathe oxygen in aerobic respiration, and we also eat carbon-based compounds. Lots of microbes can do that as well, but a lot of them can do other things. They can respire sulfur, they can respire sulfate, they can respire nitrate and nitrogen-based compounds. They respire and produce things like methane and carbon-based compounds, and they just have a whole host of other things that they can do that kind of the organisms and things that we typically think of as, eukaryotes or mammals and stuff like that can't do. We would not be able to survive on methane, for example, or sulfur, although it may smell funny to us. Not only do the microbes have this incredible diversity in metabolisms, which is encoded for in their genetic diversity, but they're just incredibly diverse overall. And so, human to human, we're like 99% related, even though we have kind of this, the same. All have arms, we all have legs, we all have eyes. But a microbe, even though they might look very similar to their neighbor, they might only share 10% of their genome as opposed to 99%. So there's just a whole host of genetic diversity of that microbes have to offer, a lot of which we don't even know yet because we haven't been able to sample them enough. If we can't grow them in a laboratory, it can be somewhat difficult to get a genome of a microbe. But in the last 20 years or so, we've really come up with better, improved, high throughput methods to study the genetic diversity. Ways to study genetic diversity have included sequencing the genomes of microbial cultures. And so that's taking a lot of microbes and extracting their DNA, and we can piece together kind of what that genome sequence looks like. We can also do a technique called metagenomics. And so metagenomics is especially powerful for environmental microbiology, looking at microbes or really anything else in an environment where it takes the DNA that's present in an environment. It kind of, when you extract it out of a certain sample, you kind of mix it all together. But with metagenomics, we can piece that back together based on abundance and also kind of, does this sequence match up with this one? And we can align the sequences together and get a better picture of the entire circular microbial genome. We can also do things like single cell genomics, and that involves pre-separating out individual microbes or eukaryotic cells. It also works really well for that. But for microbial cells, we can also do single cell genomics, separate out individual cells, amplify the DNA of just that one cell, and then sequence that DNA. And so there's a lot of these different techniques that we can now get at really good results for genetic diversity of what microbes are in the environment, but also to get a sense of the kind of, I don't want to say, microdiversity of a microbe. So really getting the cell-specific information from a microbe. And so that's really taken off in the last 10 years, I would say.
Awesome. Thank you. And then this is kind of going backwards a little bit, but can you give a brief explanation of what a gene is, what a genotype is, what a phenotype is?
Yeah, no, exactly. So genetic diversity really should really be the result of the genotype. And so the genotype is really just referring to an organism's genetic makeup, the specific genes it carries. So just to go back to the breathing example, the respiration example, humans contain genes that allow us to respire oxygen. So a gene for a protein that takes oxygen and uses it as a terminal electron receptor. And so that is what is encoded for in the genome. That's what our DNA carries through from one generation to the next. And that's where we get RNA and proteins expressed from that DNA blueprint. So that those genotype blueprints are the organism's genetic makeup, and they encode for those proteins, which then result in phenotypes. And so phenotypes, as opposed to genotypes, are the observable traits or characteristics that an organism has for it. So a common phenotype to think of is like eye color or body size or cell size and stuff like that. So it's really these physical characteristics. Another really important phenotype, both for microbes and larger organisms, is what is that organism actually capable of doing in the environment. And so all of those different metabolisms, those very diverse metabolisms that I was talking about in the last question, the ability to respire oxygen, the ability to respire sulfate, to make methane, those are all really important microbial phenotypes that come from those genotype blueprints that the organism has the DNA sequence for. So that link of the blueprint genotype to the expressed phenotype of what the organism is actually doing or expressing in the environment.
What does the process of collecting the samples of sediments to then study in the lab look like for you in your work?
Yeah, great question. And something that I've gotten really into in the last couple of years. So I would say there are three methods that I currently use to collect sediments. And it all has to do with where the sediments are specifically. So the first introduction I had to marine sediments was in the tidal area very close to Bigelow. And so we just drove a car up to where people go digging for clams and stuff. And so there's a nice little parking lot. And we just walked out into these mud flats, and we were able to take a PVC pipe, push it into the ground, cap it, and then pull up a 20 centimeter core. We were able to subsample the sediments in there, and then also, of course, the microbes that live in the sediments. Another way of physically capturing the sediment is on a smaller ship. And so Bigelow is lucky to have the research vessel Bowditch. That ship we have recently outfitted with a 2 meter long sediment core. And so we take this, 40-foot vessel out into shallow water. And I'm saying shallow, but really it's anywhere between a couple meters to 100 meters of water depth. And we drop this metal tube, which kind of looks like a lawn darts or something like that from your childhood. Maybe you throw it up and it lands and it looks kind of like a missile. And so we plunge that down into the sediment and we pull it back up, and it captures like a 2 meter long sediment core, which we can chop up on the back of the boat and subsample for the microbes that live there. And then finally, I would say the last method for reaching really deep ocean sediment is with a dedicated scientific ocean drilling vessel. And so I've been fortunate to sail on the recently retired drilling vessel JOIDES Resolution, and that is part of the Integrated Ocean Discovery Program. It used to be called the International Ocean Drilling Program. And that ship is able to access much deeper, much more sediment. It's basically this floating drilling platform. And on the expedition that I was on, we went through 2,000 meters of water column, down 1,000 meters into the sediment, and even beneath the sediment at the bottom of the water column, there is basalt and hard rock. And so we also drilled that. And so those sediment and hard rock samples are a little bit harder to get, but it's well worth it to see what the conditions and the microbes are actually present in sediment. Once we have the sediment samples, the workflow kind of becomes very similar. So these different methods of actually retrieving the sediment, they all funnel into I would say three methods of actually dealing with the microbes in the sediments. One of those methods is to culture the microbes. We can try to mimic what conditions are present in the sediment: high pressure, low carbon sources, low nutrients, or we can give them more nutrients if we want them to grow. And so we can try and grow them in the laboratory to study what they're actually capable of doing. Another method of looking at microbes in the sediment is the metagenomics that I mentioned before. And that's just grinding up all the microbes in the sediment and getting their little pieces of DNA out and kind of putting that puzzle back together of what the genomes look like. And then the third method is one that I work on at Bigelow, or that I work with at Bigelow, and that involves flow cytometry and single-cell genomics. And so with flow cytometry, we can separate out cells from the sediment and capture them in individual wells. And so the flow cytometer kind of lines up each individual cell particle and runs them through a machine. We're able to see the size and condition of the cell that runs through the machine, and we can also put them into a 384 well plate, individual cells in each well, and then multiply the DNA present in each of those genomes and sequence it for single cell genomics. And so those are kind of the three things that, as a geomicrobiologist, I focus on when looking at sediment. These things come from different methods of getting the sediment itself from the ocean.
Awesome. Correct me if I'm wrong, but you mentioned one of the ways that you study the microbes after collecting them is to recreate the conditions that they were in originally.
Yeah.
What do those conditions look like with different samples? Like samples from shallower environments versus deeper sea environments?
Yeah, exactly. So microbes, as diverse as their genomes are and as diverse as their metabolisms are, that is matched by the diversity of environments that you can find them in, which we kind of alluded to before. But even in sediment, the microbes experience many different nutrient levels, different temperature, different pressure. And so there's things in the laboratory that we can provide for them in their environment or with the food that we feed the microbes to mimic that a little better. So a lot of my PhD work actually had to do with hyperthermophiles, and so microbes that lived at temperatures 85 degrees Celsius and above, close to boiling temperatures. And so those microbes we keep in basically an oven that sits at 85 degrees Celsius or 95 degrees Celsius even. They like to live in the heat. They frequently, when they live in the heat, they eat things like sulfate and hydrogen, so we can give them those kind of not typically thought of as uh, food for microbes, but that's what they like to have available for sources of electrons and reducing and oxidizing power, sources of energy, basically. So we can mimic those really well. Something that's a little harder to mimic in the laboratory, especially when thinking about the deep sea, is the immense pressure that these microbes experience. A lot of the bacteria and archaea that I work with are kind of okay with being taken out of the deep pressure, deep high pressure environment. But honestly, since we're removing it from the high pressure environment, we don't know what we're losing in terms of like microbial diversity. There are other labs out there that have like high pressure growing tanks for these microbes, and that's really how we can see what these organisms are capable of doing and how they have adapted to the high pressure. So that's just some examples. The high temperature, high pressure, other environments that microbes can live in, not necessarily ocean based, is really high salinity or really cold. And so microbes can function at less than freezing temperatures, which is kind of cool if they have the right fluid chemistry surrounding them. And so these extreme environments, one of which is the deep ocean and the deep subsurface ocean, those can really tell us a lot about the microbes that live there. Why are they living there? But yeah, it's cool to try to mimic them in the lab. It results in some interesting setups for growing these things. Yeah.
Yeah, that does sound really interesting. And I can't lie, the whole time you were talking about that, I kept thinking back to volcanoes.
Yeah.
Just like they live in some extreme environments, but not volcanoes.
I use that as an example because I think somebody once asked me, or because I was giving a talk somewhere and I was emphasizing that we have found microbes in the atmosphere up until the edge of space, and it's hard to sample beyond that. We found microbes, 6 kilometers beneath the surface of the Earth. So where can't you find microbes if they're everywhere? And I had to think a little bit about, okay, what are the actual physical processes that would be disrupted? And the current limit that we have for the knowledge of where life can live right now is 121 degrees Celsius, temperature wise, I should say. Temperature wise, we've only demonstrated that microbes survive at 121. Why this cutoff of 121? Because 121 degrees Celsius is what we can maintain in laboratory temperatures. And so that's the only way that we can prove that they are growing. And it's one of the only ways we can prove that they're growing and living and potentially reproducing. But when you think of like, okay, what physical processes or metabolic processes do microbes need in order to live? And one of them is liquid water and the ability to get things in and out of a fat lipid membrane, basically fat lipids. And so those lipid membranes, those become disrupted when you can't maintain liquid water and when you have really extreme temperatures. And so one of the very few environments where that is a characteristic of the environment is in like an active volcano with molten rock and stuff like that. So it's just one of the very few earth-based systems that I could think of that would preclude microbial life. And yeah, there's not a lot. Microbes are everywhere. They're super resilient. They're able to adapt. They've had 3.5 billion years to adapt to anything that the planet can throw at them.
Very cool. What roles do microbes play in global processes like the carbon cycle? And can you give us an introduction to what the carbon cycle is as well?
Yeah, of course. So the carbon cycle. So there's I would say there's multiple different carbon cycles, but I'll focus really on, I'd say, two main ones. And one is particularly for the ocean, there's the biological carbon pump. And so the cycle of how CO2 is taken up by phytoplankton in the ocean, respiring microbes in the ocean also help mediate some of this. But that CO2 then kind of makes its way up the food chain to zooplankton, microzooplankton, other phytoplankton, and other predators and stuff once you get beyond like the small microbes, bacteria, archaea, and viruses. Those things can, form these aggregates and detritus of sinking particles. And a lot of the carbon cycle within that ocean is kind of the consumption or decomposition of then these aggregates of photosynthetic microbes and algae and seaweed and stuff like that. That carbon typically will fall within the water column. We like to call it marine snow. And if you send a camera down to where there's no light and you have like a little spotlight on it, it really does look like snow, just these particles constantly sinking. And so that's happening beneath the photic zone around the ocean everywhere. And so there's just a ton of material just dropping to the bottom of the ocean. And so in a lot of, diagrams that you might see of the carbon cycle, a lot of carbon does get cycled within that photic zone specifically, especially with regards to CO2 and that atmospheric interaction with the water. But that carbon ends up sinking to the bottom and it becomes buried in sediments. And so that's what I would say one of the most important carbon and nutrient cycles is on the planet right now, especially when thinking of like things like CO2 sequestration, when we're thinking of burial within sediments. That is the biological carbon cycle. Another very important carbon cycle is kind of a carbon cycle on geological scales. So in addition to kind of this biological carbon pump, there's also the deep carbon cycle. And so this is a much slower carbon cycle and it occurs on geological timescales as opposed to biological timescales. And so when I say geological timescales, I really mean millions to billions of years of time. And so this is also a really important carbon cycle. And it's even important when thinking of microbes, and especially it's important when thinking of microbes in the subsurface. And so that carbon that sinks to the bottom in this and becomes buried in the sediments, that sediment, because of plate tectonic movement, can then become subducted into the mantle of the earth. And so it takes that sediment that has become basically as hard as rock and it buries it even deeper within the earth. And so that is a really important carbon cycle to think of as well. I think is in particular important when thinking of really long-term carbon sequestration. And so instead of burying it in sediment, and when you bury something in sediment, it's not truly buried until it goes through that really deep carbon cycle into the mantle of the earth, because things can bioturbate it, things mix it up and stuff like that. So it's really just thinking on a bigger time scale of what that carbon cycle might look like, not just the biological carbon cycle, but also where does it go in terms of sediment and geology in general.
Great. And then the second part of that question is, what does that have to do with microbes and how important is that connection between the two?
Yeah, so microbes are responsible for many parts of that carbon cycle. And so especially when thinking about the microbes that are able to take up CO2 and incorporate carbon into their cellular structures, microbes are really the ones that are mediating a majority of that process, much like they do for other biogeochemical processes, but definitely with CO2. Microbes can also produce CO2, so that's an interesting kind of a secular component to the carbon cycle. But in general, they're really the ones that are mediating most of the chemical change that we see, not only in the surface water, but also the burial of carbon in sediment in particular. So they're responsible for a majority of that, I'd say.
So related to that, microbes could also play a role in degrading pollutants. Mm-hmm. Can you talk a little bit more about that and maybe what we don't know about that?
Yeah, that's a great question because we don't know a lot about it, and we would like to know a lot more. And I think we're just getting to the point with some of these, especially genomics technologies, that we can tell what an organism can do and potentially get that organism into culture to see what it can do. So just as one example particularly for the degradation of pollutants in the environment, we found a really active species of Chloroflexota. So this is a lineage of bacteria that can dehalogenate halogenated organic compounds. So these organic compounds are typically pollutants, but these organisms can mediate the destruction of these pollutants. Something that we're really interested in at Bigelow is what is the fate of forever chemicals such as PFAS in the environment? And so could these microbes potentially, when they get desperate enough, do those pollutants and these forever chemicals start to look like something that could be good for them to eat, something that could give them energy, especially in these lower energy kind of sediment environments where the more energetic compounds and the energy available from these chemical compounds has already been extracted from these longer term ecosystems. And so we really don't know if microbes can do that yet. Theoretically, they can based on other work, especially growing microbes in the lab. So we're really curious to see what the microbial phenotypes of pollutant degradation are in sediment environments, especially given that their genotypes, those blueprints, seem to indicate that they could do these things. So it's a largely unexplored metabolism, especially with regards to PFAS, but the subsurface is a cornucopia of unexplored microbial biodiversity and unique metabolisms. And so who knows what they could be doing down there, and what could they be doing today that could help us with a problem like forever chemicals and stuff like that.
Yeah, that definitely seems like a big topic that can be really important, especially with a lot of the changes that are happening in our world over the last however many years and are projected to continue as humans keep being here.
Yeah, exactly. They keep producing them, and who's going to be responsible for cleaning up the mess? Probably not going to be humans at this point.
Yeah. Thank you to Chopping Block Soaps for sponsoring this episode of Shellphone. This family-owned business makes my favorite soaps out there. They are dedicated to using high-quality, natural ingredients to create their handmade soaps and skincare products. One of my favorites is the Utah. This soap is made with shea and cocoa butter, coconut and olive oil, cedarwood and lavender essential oils, sage, beetroot, water, and lye. That's it. To browse Chopping Block Soap's selection of products, go to their website at choppingblocksoap.com and use the discount code Shellphone for 20% off your order. Awesome. So you mentioned earlier that your work also has applications or potential applications to looking at microbes in space environments. So I'd love to dive into that. And can you just tell us a little bit more about that and what that might look like?
Yeah, absolutely. So again, and this question is really what's driving a lot of my research because of my interest in astrobiology specifically. So I'm going to get back, I'm going to touch just briefly again on the respiration topic that I've been talking about a little bit. And so respiration, for you and I, it's oxygen respiration, that aerobic respiration. And so what that really means when we get to cell physiology is using oxygen as an electron acceptor. Oxygen is really great at taking on that electron from the biochemical processes, and that's what we use as our terminal electron acceptor. Other microbes, the huge diversity of metabolisms they do, have other things that are this terminal electron acceptor, so like sulfate and nitrate and stuff like that. But really, where my work is focused on currently with astrobiology is electron transfer detection. And so that electron transfer detection is really what respiration is. It's what we do when we our mitochondria and our cells do and microbes do to get energy. And I think that this electron transfer physiology of a living cell is something that is universal to life on Earth. And so if we're looking for life not on Earth, could life not on Earth also be doing this electron transfer physiology, given that life needs energy to grow, to reproduce, to move around, to metabolize, and anything that something that's defined as life needs to be able to do? So there's a famous quote: life is nothing but an electron looking for a place to rest. And I really think this is a key piece of biosignature evidence in detecting life elsewhere on other ocean worlds. And so a lot of my work currently is funded by the NASA exobiology program. And with this work, we're really interested in using different omics techniques and flow cytometry techniques to come up with these life detection strategies for low biomass subsurface environments. It really combines a lot of the key parameters for looking for life on other planets, not on Earth, including, does it look like a cell? Is it the right size? Does it have DNA, blueprint for making proteins and stuff like that? And then what I think is probably most important for my work in these extraterrestrial life detection purposes is, does it transfer electrons? Are those cells actually actively metabolizing? Are they getting energy from their environment with these different chemicals? And so my research, especially when using flow cytometry and single cell genomics, is focused on, does it look like a cell? How small can we detect cells? And does it transfer electrons? Is this technology that's deployable in an environment that might look like an environment in the subsurface of Mars or the subsurface ocean of Europa? So that's where a lot of my work, especially in the deep subsurface, is focused on with this electron transfer life detection purpose. Yeah.
When you say, is this deployable in that environment, do you mean doing the actual lab work in a setting that's not here, like in the lab that you use today? Or do you mean applying it to the microbes or the cells that you would find in those?
Yeah, so the question of, is it deployable in a specific environment or on microbes that we collect? Those are two facets that are applicable to multiple NASA missions. So for just as one example, there's currently an ongoing mission, the Mars Sample Return mission, where they're hopefully going to collect samples and finally return them to Earth. The techniques that I use with flow cytometry, staining individual cells for respiration rate phenomes, and do they transfer electrons, and then that goes into the single cell genomics of what is the cell's genome blueprint. That is a technology that is really easily applicable in both a remote environment, but also on small amounts of samples. And so if we did get samples back from the Mars sample return, we could deploy this technique of flow cytometry and single cell genomics to see, do they have that ability to transfer electrons? Does it look like a cell? Does it have DNA? So another aspect of this work is actually manipulating samples remotely. And so a lot of the deep oceanographic work uses remotely operated vehicles. ROV Jason is the one on U.S. ships on the UNOLS fleet. Yeah, so on the U.S. operated ships, they have this little robot. It's like blue and yellow colored, and it has these arms, and it's very sophisticated. It's able to dive really deep. And so we can manipulate samples remotely with this technology. And so we can develop ways to deploy a signal in that environment to detect this electron transfer physiology. And so doing it remotely on Earth is the first step before doing it remotely on another planet. So I think having that technology deployable in the most extreme environments on Earth that humans can't actually go to, but a robot could go to and manipulate, is why NASA's interested in this life detection work as well.
Cool. Thank you. I do find that really interesting. We've talked about in some past episodes using model organisms to study other organisms.
Mm-hmm. Yeah.
And this seems like kind of the same realm to me, using a model ecosystem to study an ecosystem that's harder to study.
An ecosystem that we haven't even been to.
Yeah, or that even.
Yeah, exactly. Yeah, we like to call them analog environments. So they're analogs to other worlds.
Very cool. Yeah, the episode that or one of them, we've talked about it in a couple, but one was talking about using Gulf toadfish to see how acoustic input affects them and then apply that to how it would affect dolphins. So
cool.
When I heard about that, I was like, that is amazing.
Wild. Yeah, that's awesome.
Yeah, so it's been stuck in my head, and that's why I kind of am making the connection here. But yeah, that all sounds really awesome. And I don't think I would have imagined in my head that your project existed, and I'm so glad I came across it and got to learn about it.
It's really interesting work, and I think it not only has applications to thinking about like where does life exist elsewhere, but it really allows us to better understand the capabilities of our own planet and what microbes are facilitating here. So it's kind of like you look up, but then you look within at the Earth, and we can really better understand this foundational geomicrobiology of the Earth.
Yeah, it does sound like a really important thing to get more understanding of, considering how universal microbes are.
Yeah, a big question in microbiology and microbial ecology has always been like, what is the contribution of a single cell to the carbon cycle, to the sulfur cycle, to the nitrogen cycle? And so, with these new omics technologies, we can now finally start to answer those rate questions of like, how much are they actually doing in terms of their phenotype, as opposed to just be like, oh yeah, that thing can probably sequester CO2 or something like that. It can probably uptake CO2, but who knows for sure. But now we have a lot more evidence of those actual processes.
Do we have a bit of an answer to that question now of how much one cell contributes to processes like that, or is that still in the works?
I mean, it's a little bit of both. So far we've deployed this flow cytometry and single cell genomics electron transfer physiology measuring method to the surface ocean, to the really shallow sediment in a mud flat, and also to the deep terrestrial subsurface. We've deployed it remotely in the deep oceanic subsurface, and so that's kind of working up now. And we do have a better idea of like what is the contribution of a single cell, but interestingly, across all of these environments, the answer is consistently it's a huge range. There's not really a consistent contribution of cells even within the same environment. They just one cell is really cranking it out, and the other is I think I'll take a nap today kind of thing. Like I'm not really feeling up to doing anything. So it's really different between each cell. It doesn't really depend on the cell size or cell lineage. So really, we just have more questions at this point, which can't be too shocked by that, I guess. But ideally, and we are doing this, we're going to sample a lot more to get a better sense of the even more vaster than we thought diversity of what cells can do.
Okay. So I feel like we've hit a lot of great points in a whole range of topics. Is there anything left that you want to make sure that we touch on or get to talk about before the end of the episode? Oh, wait, I did think of one kind of silly question. Earlier, at the very beginning of the episode, I asked you what microbes are. So they're microorganisms. And in doing my research for the episode, I learned that can include like bacteria, fungi, things like that, potentially, if I'm remembering correctly.
Oh, yeah.
So the silly question that I just thought of when you were talking about how some microbes don't really do too much with those processes and some do a lot, would that be due to one individual does a lot and one individual doesn't? Or is it more like this group does a lot, this group doesn't? And the silly part of that question is, are there like species of microbes, or is that not even
Oh, for sure. Yeah.
a thing?
So it's so funny. I just had a debate with someone on this. We were actually talking about the classification of planets and like what is a planet, what is a dwarf planet, and it's honest and like what is the cutoff for that? And it's kind of like a human-made cutoff of size in my mind anyway. There's probably some statistical significance to that difference. But microbial species is another one of those kind of and species in general, especially with the advent of modern genomics, species can be thought of as like if something is 97% related in the DNA sequence, it's the same species, especially for a microbe. But that hasn't always been the case. Species used to mean something else. But definitely microbes have species, they have subspecies, the kingdom, family, class, order, family, genus, species. I got that wrong. I'm so sorry. I'm a bad biologist. But they definitely have the same sort of classification scheme, but it's maybe a little different when thinking about like functional diversity as opposed to genomic diversity. Sorry, I'm trying to remember the first part of your question.
No,
Sorry.
that
Yeah.
kind of answers it. I
Yeah.
mean, the basis of the question was just what classifications exist
Oh,
within
yeah.
the group of microbes, and you answered that.
So yeah, some of the microbes do a lot and some of them don't do a lot in comparison. And so for the surface ocean, we actually found that there was a really abundant group of microbes that doesn't do a lot of oxygen respiration, but there was a specific group that did a lot, even though they were less abundant. So they represented, less than 3% of the total abundance, but they did 40% of the work. So I don't know if that sounds familiar to anyone in a workplace, but so there is definitely that dichotomy in the surface ocean. However, when you get into the sediment and the deep terrestrial subsurface, it can change a little bit. There was less of a pattern on who was the most active in terms of the species level or the lineage designation level. And so it's harder to tell other than that one surface ocean experiment.
Good to know.
No, it's a great question. Yeah, because previously the kind of the major thought, and this really fed into a lot of the models of global biogeochemical cycles and determining how much chemistry was mediated by microbes, is that the more abundant microbial species were responsible for the majority of the work. But that is not the case. It's not always the case, at least.
Great. Well, those are all of the specific questions that I had for you. If there's anything else that you want to make sure we touch on, now would be the time to do that. If not, then that's totally cool.
Yeah, no, I think we covered a lot. It is a lot of kind of disparate things coming together, but I think it's one of the beauties of doing interdisciplinary work too, is kind of like, okay, so what does this microbiology have to do with geology? And it turns out the microbes and the subsurface are completely reliant on the geology. But then it's like, what can this microbiology tell us about astrobiology and chemistry on other planets and stuff like that? So it's just a lot of links to a lot of different fields, which is good. And we need those links.
Yeah, that's a big theme that also comes up a lot on the podcast, is that a lot of topics are way too complex to just go through in an hour and completely cover everything or be like a linear progression. But yeah, thank you so much for joining us today. And I loved learning more about this. And it was great to talk with you.
Yeah, thank you so much for having me. Yeah, it was great to chat. And yeah, fabulous questions. Yeah.
Thank you. I was going to say, I also love that your name's Melody and my
I
name's
know.
Harmony.
Oh my gosh. I thought about saying something in the beginning. I'm like, no, I'll hold on. No, it's perfect. Yeah, that's so funny. Yeah, I saw that and I'm like, nice.
Yeah. Thanks for tuning in to this very sciencey episode of Shellphone. To learn more about our projects, suggest a guest, or donate to the show, visit our website at breachthesurface.org or our Instagram at breachthesurface and at Shellphone Podcast. To learn more about Melody's work and all of the very interesting work that Bigelow Lab does, visit their website at bigelow.org or their Instagram at bigelow_laboratory.